we have a brief idea about how the nitrogen pollution and the problematic nitrogen is, and also how lichens can be used as bioindicators to assess the air quality and the nitrogen pollution. So let me go through my presentation. My presentation is about briefing about how our study had contributed to study the air pollution indicators using as indicators. So this is the content of my presentation. Basically, it has three segments. So first I will go through about brief introduction about our study, and then as the second part, what we have received from Dilmah, the support we received from Dilmah, and also as the last segment of the presentation, the achievements of our project. So our study has done in the Rilagala Conservation Forest, British, close to the Dilmah Climate Center at the Navalapitiya. So the forest is located at Navalapitiya area, basically bordering to Kandy and Nuwara Eliya districts. And the extent of the forest is 209 hectares, and it is surrounded by intensively managed tea cultivation, tea plantations. So basically, the closest access to the forest is the Queensberry Estate of the Kahawatha plantations of Dilmah. So we have identified a sampling area which is close to the peripheral area of the forest, the Rilagala forest. We have demarcated a sampling plot for this study. So the extent of the sampling plot is 120 meters in length and 40 meters in width. So we have divided that sampling plot, the main area, into 5 meter by 5 meter sub-units. So it is basically, we divided it to the easy to sampling. And so we have 192 subunits in our sampling plot, and we have selected a concentrated area, the middle part of the sampling plot, for the experimental process. So we have 60 meter by 20 meter area for the experimental area. So we have 48 subunits for the experimental area. And also we do have control plots. So we have 11 control plots at the very margin of the main sampling plot. So here you can see this diagram will show you how our plots lay out in the sampling area. So at the middle of the main sampling plot, we have established a meteorological tower and the ammonia enhancement system. So Ajinkya has explained you about the meteorological tower, and also we do have a ammonia enhancement system at the middle of the sampling plot. So the system is 20 meter in length, and it will emit ammonia to the environment. It is bidirectional emission, and also the emission is it is related to the wind speed and the wind direction. We have done a preliminary survey before we start the experiment. So we have identified the trees which has 5 centimeter above DBH for the future measurements. So we have measured the DBH of each tree within the 120 by 40 meter area, and we have tagged those trees with the unique number. And we have measured the lichen, we have monitored the lichen which are on those trees. So as I mentioned earlier, at the middle of the main sampling plot, the experimental area, we have measured the lichen which is covering from ground level to 2 meters in height, and we have recorded them for the long-term monitoring. And also we have measured the canopy cover of each tree we are using for measure lichen diversity. And also we have measured the tree height. So we have measured the tree height using the pole method, and also we have used the densiometers for measure the canopy cover of each tree. And this is how our ammonia enhancement system looks like. So it is consist with it has 3-level emission. So it is made with PVC tubes, and it is connected to the ammonia cylinder, and ammonia cylinder get the signal from the meteorological tower, and it the signal will release the ammonia to the pipe system, and it will mix with the ambient air, and the mixed air will be released to the environment, and it will increase the concentration of the surrounding area. And also we are using alpha samplers to measure the ammonia concentration at each identified locations. So we have a set of alpha samplers within our sampling plot. So we can get get an idea about how ammonia dispersion happened within the plot. And then we started the lichen monitoring. So initially we have done a main survey, preliminary survey. As I mentioned you earlier, we monitored and we recorded lichens from ground level to 2 meters in height, and we have selected some lichens for long-term monitoring. So altogether we have selected 169 lichens, lichen thalli for long-term monitoring in the experimental area, experimental 48 subplots, and also 48 lichen thalli from the control subplots, 11 control subplots. So you can see here in the middle picture down here, we have tagged our lichen thalli. We have tagged our lichen thalli using ribbons and thumb pins for long-term monitoring, and it will help us. It had helped us to identify each lichen thalli with the time series intervals. And we have used a transparent sheet for measure the thallus area. So it was a quadrant divided into 1 centimeter squares, and we have put the quadrant, we have printed the quadrant on a transparent sheet, and we put that sheet on the lichen thalli and counted the cells which are covering by the thallus. So then we can get an idea about the thallus area, and we have done these thallus area measurements in monthly intervals from April 2022 to March 2024. And also we have done the lichen transplant experiment. So we started the lichen transplant experiment in January 2024, and we did it for 3 months. As Dr. Gautami mentioned you, we did it in six sampling intervals and 5 locations. Now I'll move to the second part of my presentation. This is about the support I got from the Dilmah for my study. So it was started from the I can't say that it was started from the start of my experiment. It was very far beyond that. So we started our experiment in 2021. So before that also the University of Peradeniya and Dilmah has the relationship, but I got the support from the 2021 June. So I started my plot demarcation in 2021 June. So since then I receive support from the Dilmah. So basically they hosted me. They not only me, our whole team. They host us. They provided the food, accommodation, and all our logistics. And also they have allocated a team for our fieldwork, our project. Especially they have given us a field assistant and a security for our experimental plot, and the equipment, and also the tower protection. So I should mention a name here, Mr. Vijay. He was working with like 2.5 years now, 3 years now. So he was working and giving So he was giving us all the time, he was giving us full support for the project. And also, Dilmah has given us the field assistant and the labor when we needed. In these pictures, you can see that when we need to take our equipments and gas cylinders and all our heavy material to the sampling plot, Dilmah has given us allocated the labor from the Queensberry estate, and they have supported that very much. So, and also I should mention that we have a step, for our tower, the meteorological tower, we need continuous power supply. So Dilmah has given us the CB power supply to our sampling plot on their cost. So I should mention that also. Then I'll move to the third part of my presentation. It's the achievement, current achievements. So we have identified, as Dr. Gotami mentioned, we have identified about 150 lichen species from the sampling plot and the surrounding area. So it is very rich, diverse area, and also we have recorded very good lichen, but significant lichen responses for the ammonia enhancement. So you have seen these images earlier also. So these are some responses we have monitored and we recorded from the from our experiment to enhance ammonia concentrations. This, we can use these data, we can use these observations and records to calculate the ammonia concentrations for, and also identify the critical levels for ammonia, critical levels of ammonia for lichen species. It's because the responses of lichens for the ammonia concentrations are species specific, and also they show a good response for the ammonia enhancement, enriched ammonia concentrations. So this information will be helpful for further policy making and further research. So it's the end of my presentation, and thank you very much, and also another thank you for Dilmah for supporting us.